Okay, good afternoon to everyone. So I think students from Bayombong will not be able to join us due to the power interruption. Now we'll give 5 more minutes for the other students who are from outside Bayombong to join. Baka may mga kuryente naman sila. Nonetheless, I will be recording the lecture today. So in case you are interested to watch again or to in preparation for your exam, you want to review, you can just easily go to the link that I'll send later on. Okay? Before we start, may I just ask Alisa, Luis, Miss Domingo, can you lead us into a simple prayer please, so that we can start the class today? Thank you. Amen. Thank you very much, Miss Domingo, for that short but very meaningful prayer. So welcome to our second to the last meeting. So we have, excluding this, we have one actual or formal lecture meeting left next week, and then you are off to your final exams. Okay, it's been a challenging semester as we are still in the pandemic, but uh, we have proven that we are stronger and better than this pandemic. And until this time and the coming weeks, we will uh, persevere. Okay. Now, before we start today's session, so let me just share to you my screen. I've just discovered this, and um, probably this has just been recently added. So can everybody see my screen? Can you see what I'm sharing?
Yes, sir.
Okay. So during the lecture, at any point, if you have any question and you are hesitant to ask your question verbally in class, you have this option here. Okay, if you look into the right corner or right side of your screen, you have here a portion called Q&A. Okay, so this is a feature of our Google Classroom that allows you to ask questions from me or even from any of your classmates. Okay, you can answer actually if you see the questions being posted. So just click the Q&A part of your screen. And then you have here in the lower portion a plus sign that says ask question. So again, as I've said, if you are hesitant to ask your question verbally, okay, in class, you can also type it. You can write your question, type it in here. Just click ask question, type it. As soon as you type your question, just post it there, okay? After the class or during the class, if I have a chance, I will answer your question, and then others may also answer or post their response to your question so that we can make this collaborative and the same time participative in nature. Okay? So please use this feature anytime you like, especially if you have query or if you have something to clarify, okay, and you don't want to speak or to say it in the online class, then you have an option to type it here. Are we clear? Okay, wonderful. Okay, we have others still who are joining. So, we have been discussing in the past 3 weeks the biomolecules that we measure or that we assess or uh, evaluate in clinical chemistry lab. Now, these biomolecules that we have been discussing are, of course, your carbohydrates, specifically glucose. Okay. And then we went last meeting on lipids and lipid measurements. This time, we focus our attention to proteins and other related substances. Okay. Now, I have uploaded in your um, LMS. You can still see my screen, right? Can you still see my screen, ladies and gentlemen?
Yes, sir.
Okay. So if you go to your learning management system, down here on the portion labeled learning materials, you have the folder labeled as proteins. Click on this so that you will open the 3 videos that I have uploaded, which will help you understand more protein analysis that we perform in clinical chemistry lab. Okay? This will help you understand better class discussion, especially if you learn best by uh, hearing at the same time seeing illustrations. Okay? By viewing videos, you are able to absorb more the concepts, then these 3 videos will really be very helpful. So please go to this later on after the class. And I will also be uploading here the presentation and the lecture notes. Okay? Are we clear? Maliwanag.
Yes, sir.
All right. That being said, let's proceed now to the lecture material. Let me just share to you the lecture material. Can somebody tell me if the presentation is moving on your side? Is it moving?
Yes po, sir.
Great. Okay. Let me just skip that for a while first. Uh-huh. And put this on recording mode. Um, Miss Balikat,
Sir?
can I make you as the co-host, my dear, and then you be the one to accept your classmates who are still joining so that I can continue with the presentation?
Sige po, sir.
Thank you very much. Okay, so let's proceed to the lecture now. Is are the slides still moving?
Yes, sir.
All right. Thank you very much. So today's main topic is all about protein analysis. Okay. Now, as you can see, the second word is in plural because when we analyze proteins, we don't just measure the total protein level in the plasma. Okay. Now, of course, the protein analysis or analysis that I'll be presenting today are analysis using blood as your specimen. I will not be covering in this lecture the proteins that are measured in urine, in CSF, okay, and in other body fluids because those topics are covered in your other courses like in your analysis of urine and other body fluids, AUBF, as well as probably in your parasitology for or microbiology for the comparative uh, feature of the different types of meningitis, for example, using the protein as basis. So my focus today would be mainly the measurement of proteins in blood, okay, with blood as your primary specimen. Now, for our discussion today, class, we shall be guided by the following um, objectives. Okay. So at the end of the session today, you should be able to describe first the general characteristics of proteins. Okay. You have discussed in detail the chemistry of this biomolecule in your biochemistry course last semester. So I will not be spending so much time discussing already what you have covered in your biochemistry. Okay, I would rely on the fact that since you passed that course, that you have learned and retained the information that was shared and taught to you by your instructor, and that you have brought those information over to clinical chemistry. Okay, as you know that courses in MedLab as well as in other disciplines, I believe, are all integrated, integrative. Informations from one course to another will help you, are all connected together and will help you understand better the concepts. Okay. So in the general characteristics of protein, I will just be highlighting the most important characteristics, which, as I've said, would be a repetition of what you have discussed in biochemistry. Okay. Second, at the end of this session, you should also be able to describe the structure and classification of proteins. Okay, especially the structure. Okay, I believe your teacher in biochemistry has walked you through um, intensively with how the structures of protein are organized from the primary to the quaternary. Okay, I will just emphasize on some of those with, of course, um, connection to those that we analyze in the lab. And then for classification, the classification scheme that we look into here is the simpler classification scheme where proteins are just grouped into two main classes. The third learning objective is for us to discuss the functions of protein in the body. Okay. Um, it is necessary that for us to really understand how proteins can be used as indicators of disease, or how proteins can be used to understand the mechanism of disease. Okay, we have to be able to first learn what roles do these proteins perform in our body. Number four, we should be able to explain the different fractions of protein in the body. Okay, so proteins exist in different form in the body. Okay, although the main form, which is called albumin, is the most abundant. Okay, there are other forms of protein that are actually as important as albumin. So we'll discuss that in number four. And in light of the application of these proteins in disease management, treatment, and diagnosis. The fifth is to identify the different methodologies used in measuring protein in biological analytes, particularly blood. Okay, so as medical laboratory scientists, this is where our job, okay, comes in. We are involved in performing analysis, and then once we have completed the analysis, we give the result of our analysis to the doctor for interpretation. Now, we are not really the ones who are involved in interpreting the results, but we do understand the result. Okay, we do understand the implication of the results of the test that we perform. Okay, however, the interpretation aspect is beyond the scope of our profession. Okay, but like what I've said, number five is the main focus of our job as medical lab scientists in the analysis. So we are masters of these methodologies, the different methods that we can use to measure activity of protein in our specimen or in patient specimen. And then last but not the least, taking the result of our analysis, we should be able to correlate the level of total proteins in the different types of specific proteins to different clinical conditions. So, say for example, a patient suffering from uh, liver failure, okay? So, how would this condition affect the result of our measurement if we do total protein albumin analysis on that patient's sample? Okay? So these are the 6 learning objectives that we will be covering in today's session. I hope that I was able to clarify to you things and show you the direction of our discussion today. Okay? Is there anything that you'd like to ask before I proceed to the next part of our lecture?
None.
Anything?
Nandun po, sir.
Okay. Now, like what I said at the start of the lecture, if you have any question and you're shy or you're hesitant to ask it verbally in the class, there is a question, a Q&A tool available for you there on your side in the Google Classroom or Google Meet site. Type your question there and I'll review those questions later on before we dismiss and I could answer those questions, or even some of your classmates can also participate and reply to your questions. Okay? Let's begin with the general characteristics of proteins. Proteins are complex molecules class. Okay? They are large, complex molecules. Their basic structural component, okay, the building block or blocks of proteins are called amino acids. Okay? So proteins are large biomolecules that are made up of building blocks called amino acids. These amino acids are joined together through a chemical bonding, a covalent bond called peptide bonds. Okay? Now, if you can recall your chemistry 1, okay? In chemistry 1, you discussed that generally there are 2 types of bonds that can be formed between different atoms of elements. Okay? One is called the ionic bond, and the other one is called a covalent bond. In an ionic bond, you would see the transfer of electron, the valence electron from one atom to another. Okay? On the other hand, when you have a covalent bond is formed when the valence electron of 2 atoms are shared together. There is no transfer, there is no movement of electron, but the electron or electrons are shared. That is what we call a covalent bond. Now, covalent bonds class are usually formed or are usually seen in organic substances like proteins or the building blocks of proteins at least. Okay? So when these building blocks of proteins called amino acids join together to form the protein, okay, the linkage that connects these amino acids, okay, that link is called the peptide bond or peptide bonds. Now, a peptide bond is formed between the carboxyl group of one amino acid and the amino group of the next amino acid. As this carboxyl, COOH, and NH2 amino group join together, we see the loss of a water molecule. Okay? Is there any question there regarding peptide bond and amino acids? So proteins are made up of amino acids. Amino acids are linked together to form proteins. Now, the type of amino acid as well as the sequence of amino acid that makes up a protein class is of course determined and dictated by the genetic material, okay, by the DNA. The genetic material in the DNA, which has been copied into a messenger RNA during the process of what we call transcription. Okay, if you would recall your cell biology, which you took in your human ana and physio in the beginning of the course. Okay, so the DNA is the molecule that dictates what type of amino acids and what sequence of amino acids are present in a protein. But I will not be going down to that more detail on protein production and synthesis because you will be covering that in a separate course. I just want to for you to at least have a recollection of that. Maybe you have taken that in your uh, anatomy course before. In a protein structure class, there will always be a part where the amino is the end, and then you call that as the end terminal portion. And there's also a part where there is a carboxyl end, and you call that as the C terminal portion. Okay. Now, among proteins basically are made up of four different types of atoms: an atom of carbon, hydrogen, oxygen, and nitrogen. Plus, in some proteins, they also have sulfur. So nitrogen is actually the atom that distinguishes the protein from carbohydrates and lipids. All proteins contain at least 16% of their structure, which is nitrogen. Okay? The nitrogenous content of protein makes up 16% of the total protein structure. Okay? Now, speaking of protein structure can be classified into four levels. We have the primary structure. If you look into the illustration on top, in the primary structure, you have the amino acids linked together through peptide band arranged in chain. Okay, so this is, if you look at the arrangement in chain, that we can describe this as a linear arrangement. Okay, the primary structure of protein is created by the linkage, okay, of the amine group to the carboxyl group of one amino acid to the next one. Okay, the linkage is of course formed by covalent bonding. So primary structure, linear structure of amino acids, or the linear arrangement of amino acids. This primary structure is formed again by the peptide bond. Okay, the bond created between the amino group of one amino acid and the carboxyl group of the next amino acid, or the second amino acid. Now, the second level of protein structure is the secondary level, or the secondary structure. Here, we now see the formation of alpha helix and as well as beta sheets. Now, what causes the polypeptide to form alpha helix structure and beta sheet structure? The alpha and beta structures class are due to the formation of hydrogen bonds, okay, between the carboxyl group and the amino groups of the peptide bond. Okay? In the primary structure, you have the peptide bond created, linking the amino acids. In the secondary structure, you now have the hydrogen atom of the carboxyl and amino group forming attraction. They link also together. So it's not just a peptide that links together the amino acids. Even the structures in the peptide group, in the backbone, in the connection, will also interact to form folding. Okay? The linear structure suddenly folds, creating a helical or a beta pleated or beta sheeted or pleated secondary protein structure. Okay? Again, I repeat, the Formation of the secondary structure is due to the interaction of the hydrogen group of the carboxyl and amino. There is a formation of a hydrogen bond. Okay, the hydrogen bond, of course, is different from the peptide bond that is connecting individual amino acids. Within the peptide bond, you can see the hydrogen bond forming, causing the linear molecule to fold to form what we call alpha helix and beta pleated structure. Now, in the third level, the tertiary, we now see here the coiling, okay? The coiling of the polypeptide chain that has initially folded, and that coiling is due to the interaction of the R group, the specific amino group or amino acid group, okay? This coiling as a result of the interaction of the R groups class creates the tertiary structure. In this tertiary structure, you now have disulfide linkages, hydrogen bonds, hydrophobic structure, and even the van der Waals forces affecting how the coiling occurs. So the molecule has not just folded to form helix or pleats, but has also coiled or has resulted into coiling due to the interaction this time of the specific amino R groups that are attached to the peptide bonds. Okay? The fourth level is called the quaternary protein structure. Here you now see two or more coiled polypeptide chains that come together to form a complex protein molecule. The ability of these identical coils to group together, to come together, is made possible by the binding of hydrogen bonds as well as the electrostatic interaction or attraction between the different components or subcomponents. Okay? Basically, you have two or more similarly looking tertiary structures that join together to form one functional protein. Okay? Like in the case given here in this example, we have hemoglobin. Hemoglobin is the protein found inside red blood cells. It is the component of red blood cells that basically gives the red blood cell its function. Without this hemoglobin, the red blood cell will not be able to perform its function. Within this hemoglobin molecule class, you have four subunits. Okay, these four subunits are organized into pairs. So there are two pairs. Each pair is identical, such that you have what we call an alpha pair. So when you say alpha pair, the two components of the pair are both alpha proteins. And then we have the second pair, which are non-alpha. Okay, non-alpha. We always designate the second pair as non-alpha because the non-alpha group differs depending upon the specific type of hemoglobin. If you are an adult, the majority of the hemoglobin that you have in your RBC is called hemoglobin A. And that hemoglobin A has two alpha subunit and two beta. However, if you are a developing embryo, okay, you have just been fertilized, you are just growing in your mother's womb, you are in the embryonic stage, okay, the type of hemoglobin that you would see in the primitive red blood cells in that embryo will not be hemoglobin A. Okay? It could either be hemoglobin goer 1 or goer 2 or Portland, which does not have the same non-alpha subunit. You'll have more of that discussion later on when you take your hematology 1. Okay? So that's the quaternary protein structure. There are other quaternary proteins other than hemoglobin here, but the hemoglobin is the what we call classic example since it's the one that can easily show the progression of the different levels of protein structure. Okay, so as a review, protein structure can be classified into 4 levels. Primary, if the amino acids are in linear arrangement. Secondary, if the amino acid starts to fold, forming helix or helical arrangement or pleats. Okay. In tertiary, we now have the uh, protein forming coils. Okay, not just folded and not just folded, but the protein has been coiled. Okay, or has coiled due to the interaction of the different components of the protein molecule. And then in the quaternary, you now have the joining of similar tertiary structures or coiled structures to form one functional protein. Okay? Is there any question or clarification?
Sir?
Hello? Do I still have students in the online class?
Sir, ako po.
Yes, what's the question?
Sir, ka-clarify ko lang po sana yung sa may R group sa
Hmm.
protein structure yun din po ba yung tinatawag na side chain?
Yes, yes. Those are the side chains. Those are your specific amino acid side chains like your lysine, glycine, glutamine, cysteine. Those are the R group. So the R group interacting together will form your tertiary structure.
Thank you, sir.
You're welcome. Did I answer your question?
Yes, sir.
Okay. Anything else? Meron pa? Okay, I'll take your silence as no more questions. We now move to the classification of proteins. So the classification scheme that I will be presenting here to you today, class, is a simplified classification scheme. Basically, the classification scheme that you have here groups the protein into just 2 groups. We have the simple proteins and the conjugated proteins. So simple proteins, basically the structure is made up purely of amino acids. Okay, amino acids that are organized into different level of protein structure. So under simple protein category, we have the globular type and the fibrous type. Okay, the globular type of protein, like your albumin. By the way, albumin is the protein here in the upper left hand illustration. So this is the crystallized structure of albumin. It's a globular protein. The structure is said to be symmetrical and that the polypeptides are compactly folded. Okay? So the polypeptide chains are not linear at all. Okay? They are not simply helical or pleated, but they are compactly folded, okay, structures. For the fibrous type, they are usually elongated, asymmetrical, okay, unequal length, okay. And the best examples given here are troponin. You have troponin in the upper right illustration. And then we have here below, the one in the middle, you have collagen. These are actually crystallized structures of the proteins. Okay, these proteins are simply made of amino acids. That's why they are called simple proteins. All right. I wouldn't really call them as well pure protein because that might be misleading. Okay. In nutrition, we also have classifications like complete and incomplete proteins, but that has something to do with the amino acid content, which I don't want to go into anymore because that's outside the scope of our course. So for simple proteins, I'd like to stick with the definition that they're mainly, when you look into their structure, you can see amino acids. Okay. Now, if you see other components other than amino acids, then you are looking into the second class, which is the conjugated protein. Conjugated proteins are protein plus another substance, hence the term conjugated. So we have metalloproteins. Metalloproteins are proteins with a metal ion in its structure. There's a metal ion prosthetic group, like in the case of ceruloplasmin. Or you can also have here in this category hemoglobin, because hemoglobin is not just purely protein. The protein part is called globin, and then the heme part it's a non-protein component that makes now hemoglobin a conjugated protein. In the non-protein part component called heme, that's where you can find the ion, the metal iron. Okay, iron. Then we have the lipoproteins. If you can recall lipoproteins, we have just discussed this last meeting. Lipoproteins are the carriers of lipids, okay? But they don't just simply carry lipids on top of them. Lipoproteins actually have a very complex, unique structure and arrangement with that of its protein part. The protein part of lipoproteins is called the apolipoprotein. Remember, you have different types of apolipoprotein. Um, APO100, ApoB100, ApoA48, ApoA1, A2, okay, or apolipoprotein A1, A2, okay? Those are your apolipoproteins, which are the protein parts of the lipoprotein. Now, for the lipoproteins, remember the lipoproteins we discussed last week, we have chylomicrons, VLDL, LDL, IDL, and HDL. Even LP, small a. Okay? Those are the lipoproteins. They are not just made up of lipids, they have a protein component, making them classified as conjugated protein or also called conjugated lipids. Then we have glycoproteins. Glycoproteins usually class are found on surfaces of cells. They act as membrane markers. Okay, they act as membrane markers, or sometimes they also play what we call antigenic properties or antigenic functions. Glycoproteins are proteins that contain about 10 to 40% carbohydrates. Okay. One example given there is haptoglobin. Haptoglobin is a conjugated protein which binds with hemoglobin in the circulation. If you have red blood cells undergoing lysis, your RBC is hemolyzed inside the blood vessel, the lysis results to the release of the hemoglobin content of the RBC. That hemoglobin content, some of it will have to bind with haptoglobin. The haptoglobin is the carrier of hemoglobin in the circulation. Then we have mucoproteins. Mucoproteins have about more than 40% carbohydrates attached to the protein. Okay, so this is a higher carbohydrate content or containing protein. Okay, so mucoproteins, the example given there is mucin. Mucin are complex molecules that can be detected in some body fluids. Okay, they well, up to the present, their diagnostic value may be very limited. That's why they are not routinely measured or evaluated in the laboratory. And then we have the nucleoproteins. Okay, these nucleoproteins are proteins that are conjugated or bound to the nucleic acids, yeah, like your DNA, okay, or your DNA or RNA. Okay, they usually exist in cells as, like, for example, your chromatin. Okay, chromatin molecule. Chromatin is that conjugated structure or molecule that is made up of compacted DNA plus the histone molecule where the DNA coils around. Okay? So these are the conjugated proteins. Okay? So two classes of protein. We have the simple protein, globular and fibrous, plus these conjugated proteins like metalloproteins, lipoproteins, glycoproteins, mucoproteins, and nucleoproteins. All right? We then proceed to the functions of proteins. Proteins generally class are known to be structural molecules. Okay? They are molecules that are involved in building structures in the cell, the tissues, and of course the organ and organ systems. Okay? The main role that they play is to become parts of the structures that make up our cell, our tissues, our organs. On top of that, of course, we have other roles that they can do like energy production. For every gram of protein, we can actually produce 4 kilocalories of energy. But in the human body would obtain energy from proteins only when there is no glucose or carbohydrate available. Okay? The primary source of energy would always be carbohydrates or the glucose. Okay? If there is no glucose or if the body is depleted of glucose, that's a time that your body would want or would start to burn now your proteins. Okay? That's the reason why in severely starved individuals, whether they have been starved intentionally or non-intentionally, if they have been uh, like starved due to famine or the insufficiency of nutrients, if you notice, these individuals, as they the starvation progresses, okay, they start losing muscle mass, bone strength, okay, because at that stage in their starvation, due to the absence of carbohydrates and even fats, because fats have been burned out already also, proteins, okay, are the next option of the body to be converted into energy. Until a point, of course, that the proteins will also uh, run out and eventually, well, the organism or the human body would just expire or die. Okay? Because, of course, without any energy or energy source, there would be nothing to fuel or energize the cell. Okay? So, yes, it's true that we can get 4 kilocalories of energy for every gram of protein that we convert. Okay? But in the body, in the human body, proteins will be the last option for the body to convert into energy. The proteins will also influence water distribution, okay, in the human body. Okay, so regulation of water distribution is one important function of proteins. Proteins class are osmotic regulators. Okay, when you say osmotic regulators, the proteins determine where the water goes. Okay, if therefore, if you have very little protein in your blood, okay, the tendency is the osmotic pressure in the blood will be very low, causing the water to actually leave the intravascular space and go to the extracellular space. Okay? Like the one that you see in patients who suffer from edema. Okay? The patient becomes edematous. The edema is a result of water going to the extracellular spaces. Instead of water staying mainly in the plasma, okay, inside the blood vessel, without the protein controlling the or regulating that water, the water would now be escaping, okay, leaking into the extracellular space, the interstitial space, the space outside the cells, causing what we call edema or swelling. Not the swelling related to inflammation, of course. This edema, especially related to decreased protein level in the blood, is a more pronounced extravasation of the water in the blood plasma. Proteins are also important buffers. As a buffer, proteins control or regulate the level of hydrogen ion available in the body, which of course affects the pH of the body. Okay? So proteins are very efficient in removing and controlling the level of hydrogen ion, or therefore controlling the pH of the body, the blood, and the overall the body. Proteins are important transporters as well. Okay? Transporters of what? Of different molecules. Like the molecules that are um, hydrophobic, molecules that are non-polar, they don't dissolve in water. Remember, class, that blood plasma, which is the liquid component of blood and makes it possible for blood to circulate in the blood vessels, okay, that blood plasma is 91% water. So if you have a substance, a solute in that blood plasma that is considered to be non-water soluble, okay, non-water soluble, how would it be transported then by that water-containing plasma? Okay, the remedy or the answer to that is, of course, the transporters, proteins. Proteins that carry these what we call non-water soluble substances, making it possible for them to be distributed around the body. Like, for example, steroid hormones or hormones that are derived from cholesterol. They need carriers, okay? Like your estrogen, progesterone, these hormones require carriers in your blood to be transported. All right? Another function of proteins is as antibodies. Antibodies, as we all know, are important components of our body defense, our immune system, our ability to fight off, okay, potentially harmful substances that have the ability to alter the balance inside our body or to alter homeostasis, which results, of course, to disease. We also have proteins that are very important in making cellular structures and as well as making possible cellular processes. Okay? So we have proteins that are mainly dedicated to performing functions within the cell to make up the structure of the cell, like the cytoskeleton, okay, and as well to be used to carry out the functions of the cell. Okay. Then we have, as I mentioned earlier, the structural proteins, the proteins that build up our cells, our tissues, our body. Okay. The proteins that basically their main function is to form the structure, to form the anatomy of the different levels of uh, body organization, from cellular to the most complex whole organism. And last but not the least, of course, proteins are enzymes. As enzymes, they have the ability to speed up chemical reactions. Okay? And as enzymes, as they speed up chemical reactions, they are not utilized. They are not changed in the chemical reaction, making them very efficient, okay, making them um, reusable. Okay, as they are not changed, they are not consumed in the reaction that they catalyze. Okay, they can be used several times. They can be utilized several times, and therefore the body doesn't have to be continuously synthesizing them. Okay, or the body doesn't have to be always creating them since they can be reused many times for a specific reaction. Okay, so enzymes are proteins. These proteins that act as enzymes are very important in many, if not all, bodily functions and processes. Okay? A lot, if not all, body processes, metabolic body processes rely on enzymes. Without these enzymes, there would be catastrophic results or catastrophic outcome in a living organism. Okay, as we have seen in many cases, even if just one enzyme deficiency can lead to fatal outcomes. Okay, those are the functions of proteins. Do you have anything to add or anything that you'd like to clarify? Anyone? Anyone from the class? Oh, wala na namang sumasagot. May katanungan ba?
None, sir.
Okay, salamat sa sumagot. We're now moving to the methodology for protein analysis. So, we will look class into the different methodologies that have been used to analyze proteins, even methodologies that have been used in the past. But I'd like to emphasize only or mainly into the methodologies that we can use actually up to the present time in the clinical chemistry lab. Okay, so we begin with refractometry. We are still using refractometry in the lab, okay, once in a while, especially if we want to check. Well, refractometer has been used for checking specific gravity of urine in the lab, but not mainly to check protein levels. But there are available refractometers in the lab, I'm sure. Even if your lab has already adapted sometimes the more sophisticated technology, if you try to look into your equipment and materials, you will still find these refractometers. Now, what is refractometry and how does it work? So in refractometry class, we measure the level of the analyte based on the bending of the light as it passes through the boundary between air and water. Okay, it usually is said to be a function of two transparent layers. Okay, now the bending in protein analysis, the bending of the light is usually proportional to the concentration of the protein, the solute present in the solution. In the solution here, of course, is the specimen, okay, the patient specimen, which should either be the plasma or the protein. The higher the solute concentration, the higher the protein concentration in your specimen, the higher will also be the angle of bending, and as a result, the higher will be the refractometry reading. Okay? Now, refractometry as a way to analyze proteins in clinical samples have not really been used for a long time already, okay? Because we now have more sophisticated method to quantify proteins. Because refractometry could be subject to a lot of interfering or intervening factors which can potentially decrease the clinical utility of the results that we obtain. Okay, but the principle is or has been laid down here. Okay, the light bends as it passes through the solution containing the solute. Now, the more solute in the solution, the higher or the larger will be the bending, the larger will be the refraction or bending of lights. Okay? So that's the first method. The second one here is called the burette method. Now, sometimes some people would read this as biuret. Okay? I don't mind whichever pronunciation you would adopt, burette or biuret. The important thing to me is you know how to spell the word correctly. Okay? So the burette method is based on the complexing of cupric ions with a peptide bond of the protein in an alkaline medium to form the final color, which is purple. Now, the intensity of the purple color is directly proportional to the amount of peptide bonds present in the sample. Now, the number of peptide bond class increases with the number of amino acids present in the protein, of course. Okay? So, it has been said that in nature, the simplest amino acid that has been detected is made up of 20 amino acids, which gives you the idea, class, that the proteins that we find, especially in the human body, they are really large proteins. They are made up of more than 20 amino acids, sometimes hundreds, thousands even. And the higher the number of peptide bonds, which is the one detected by the burette method, the more intense will be the purple color. Okay? And then the purple color is, of course, it could be read photometrically, okay, to quantify the concentration of the protein. It's like an indirect measurement because what the burette reagent detects is the peptide bond. Okay, so you could say, or you could argue that um, if your sample contains even non-protein molecules or some substances that are not necessarily proteins but polypeptide in nature but less than the number of amino acid to qualify as protein, they would still give a reaction. Okay, in the illustration that you see there in your picture, in the slide, class, you would see that the results are interpreted as either negative or positive, giving you already the idea that this burette method initially could just be considered as a qualitative method, not a quantitative method. Okay? Because when you see that the solution has turned purple, that is already an indication that your sample contains protein or sample contains something that has a peptide bond. Okay? If there is no purple color, then that means that there are no peptide bonds present. Now, the intensity of the purple color can be an indication or could give you the idea that your sample contains a lot of proteins. Okay? You can quantify this by doing photometric measurement to detect the absorbance of the solution. Okay? Particularly as, well, you have to find the wavelength to which the absorbance can be maximally detected. This time, of course, the color would be purple or violet. The third method is a dye binding method. In the dye binding method, the principle is that the proteins would bind with a dye, a colored substance, forming a dye protein complex that will shift the maximum absorbance of the dye. In this case, the dye reagent is called Comacy Brilliant Blue. Okay, so Comacy Brilliant Blue is already a colored substance. It's a dye. Once it is bound to protein, the intensity of absorbance of that dye changes. Okay, and that the intensity of absorbance change is directly proportional to the protein concentration in the sample. So this is a quantitative assay. This will allow you to measure the level. The absorbance is read at 595, okay? And we say that the absorbance will be directly proportional to the concentration of the analyte or the protein in the sample. Okay? The dye binding method using Comasi Brilliant Blue is sometimes referred to also as the Bradford assay or the Bradford method. And then we have an older technique called the Keldal technique. This one, it does not measure directly the protein, but the measurement is based on the nitrogen content of the protein. Okay? It is a very taxing method. It's a long process. Imagine if we are still doing this Keldal method now in the clinical lab, we will not be able to release our results within the day, okay? Because we have to prepare this setup depending on how many samples or how many patients we have. So it is a very taxing method. It's a tedious method, long method, okay? But it has been said based on literature that this method is the reference method of choice to validate the materials used for the burette method. Okay? So to validate the techniques that you can use in the burette method, you can use of the Keldal technique. So for research purposes, this can be adopted Keldal, but for the actual clinical lab testing of protein, we do the dye binding assay because they are more appropriately applicable for high patient volume. And then we have electrophoresis. Electrophoresis class is actively being used in clinical setting, in the clinical lab. If you don't see this being utilized in chemistry, you would see this in HEMA. Okay? Particularly if the hematology lab is offering tests for hemoglobin S or for thalassemia diagnosis, then electrophoresis is very important or is commonly seen there in the HEMA lab. Electrophoresis is both a qualitative and a quantitative technique. Okay? Qualitatively, you can use electrophoresis to separate, okay, and then also identify the proteins. So we have three types of electrophoretic technique mentioned here. We have the serum protein electrophoresis, the high resolution electrophoresis, and then we have the isoelectric focusing, which is a type of zone electrophoresis. In the serum protein electrophoresis, the basis for separation is on the electrical affinity of the protein molecules. Now, protein molecules class, depending upon the pH, at pH 8.6, protein molecules will exhibit a net negative charge. Okay? And as a result, because they have net negative charge, they will travel towards the positive side or what we call the anode. Okay? The anode. So, the movement of the molecules would be from the negative side, the cathode, towards the anode. Okay, we have to remember the rule that opposite charge or charges attract, similar charges repel. So in this case, you have in the cathode at the beginning of the electrophoresis chamber or the electrophoresis immobile phase, you have there the well containing the specimen, and then depending on how negative is the molecule, you will see the molecule migrate through the immobile phase, okay, to the stationary phase, okay, which after the whole process has been done, the separation process has been done, you develop the electrophoresis strip by staining and checking the separation of the protein. Now, it's not just the charge though of the protein that will influence the migration of the this protein in the electrophoresis chamber. it will also be the molecular weight, the size of the molecule. So if the molecules are quite heavy, naturally, they will not be able to migrate as far from the cathode, okay, from the origin. But if they are lighter, less dense, they have the tendency to migrate faster in the electrophoresis chamber. So among the proteins, the fastest in migration is albumin, followed by alpha-1 globulins, then the alpha-2 globulins, then the beta globulins, and the least when it comes to migration, those that are slowest are the gamma globulins. Sometimes they don't even move from the origin. Okay. After the separation, as I've said, the proteins are stained, they are colored, and then the results is read using a densitometer. Okay, using a densitometer to not just identify what is the protein in the sample, but also to determine the quantity of that protein. Now, there's a modified method of the SPE. It's called the high resolution electrophoresis. It makes use of agarose gel at higher voltage, plus there is a cooling system and a more concentrated buffer that will allow you to separate as many as 12 zones, okay? To separate the proteins into as many as 12 zones, making it easier to distinguish and identify these proteins. The third type of electrophoresis mentioned here for protein analysis or measurement is the isoelectric focusing. This is a type of zone electrophoresis that identifies and separates the protein based on their isoelectric point. The isoelectric point of the protein class is the point where their charge is at net zero. Okay? So that is electrophoresis. It's a method that is both used to separate, identify, qualitatively, and even quantify the proteins. The next set of methods, they are collectively called the immunochemical method. The immunochemical method class relies on one specific principle, and that is the antigen-antibody reaction. Among the biomolecules, proteins are the most antigenic. Okay? And based on this characteristic, we can isolate proteins, inoculate the protein into a lab animal. The lab animal will create antibodies, and then we can harvest the antibody and purify and use those antibodies as reagent to identify specific proteins. Now, there are many different preparations of the immunochemical methods, one of which is demonstrated here. It's called the radioimmunoassay. So in the radioimmunoassay, your antibody reagent is labeled with a radioactive molecule. Okay? So there is a radioactive molecule that is attached to the antibody. All right? So when the reaction takes place, when the antigen has binded to the antibody, the binding of the antigen antibody could be measured either by a decrease in radioactivity or an enhanced radioactivity. The one illustrated here is a more complicated method because this is not just a simple RIA or radioimmunodiffusion or radioimmunoassay, but you also have what you call a competitive binding assay. So you have an unlabeled antigen and a radio-labeled antigen. Okay. The unlabeled antigen can combine with an antibody. Okay. Once the unlabeled antigen binds with the antibody, it may display some radioactive activity, creating or which enables you now to measure that radioactivity either as an enhanced radioactivity or as a decreased radioactivity. Okay? There are other preparations like immunoturbidimetry as well as immunonephelometry, but the basic principle is on antigen-antibody reaction. The specific antibodies will only react to their specific antigen, And this antigen-antibody reaction can be used to identify and even quantify the level of protein. Okay? Questions so far before I continue with this slide here? Any
Sir?
questions? Yes.
Sir, sa ano po? Tanong ko lang sa may electrophoresis, sir, kasi hindi ako sure. Sa may, bakit, sir, bakit lagi pong sa cathode yung origin ng proteins?
Okay. Cathode palagi ang origin ng proteins class because we control the pH of the reaction vessel. So if we uh, control the pH at pH 8.6, we will be converting our proteins, our proteins in our sample into a net negative charge. So naturally, mag-start talaga sila sa cathode. Now, if the specimen contains lesser amount of negatively charged particles, then you would not expect the substances in the sample to really migrate significantly kasi nga wala namang masyadong negative charge content. So naturally walang attraction doon sa anode. Okay? Pero pag maraming masyado yung protein that has been converted into a negatively charged particle, you would see a significant migration from the cathode to the anode. The samples are applied in the cathode because the samples have been uh, programmed to be charged or to have a net negative charge. Proteins class have both what we call a negative part or negative charge component and a positive charge depending upon the pH kung ano yung mas malakas doon. I forgot the concept but they call it as the amphi I can't remember the exact term, but it has something to do with the duality of the charges that proteins can display depending upon the pH where they are found. So at pH 8.6, at alkaline pH, the proteins will be more negatively charged. Did I answer your question? So we control the pH, we set the pH at 8.6, and we convert the proteins into a net negatively charged particle. That's why we have to apply it in the cathode, which is the negative side, because the substance will have to migrate the opposite side towards the anode. We cannot apply it immediately to the anode because we will not be able to separate anymore the substance. Tayo na mismo ang gumawa ng migration niya in that case. The direction will always be from the opposite charge, from the cathode towards the anode. As we said earlier, negative, unlike charges attract, like charges repel. Did I answer the question?
Yes, sir. Thank you po.
Okay.
Sir,
Yes.
sir, yung inaalala niyo po bang term, sir, is amphoteric?
Ayun, amphoteric. Oo, amphoteric molecule.
Yes, sir.
Thank you.
Welcome, sir.
Amphoteric molecule, yes. Depending upon the pH where the protein is found, sometimes it could have a net positive charge in acidic pH, but in alkaline pH, usually it will have a net negative charge. There is a alkaline electrophoresis technique class, meron din tayo tinatawag na acid electrophoresis technique. So, it usually is dependent upon the buffer that you use. If you make use of a buffer that promotes the pH to a more alkaline pH, then the proteins will assume a net negative charge. Okay?
Thank you, sir.
Okay, let's move on to the discussion of the clinical significance of the specific proteins that we assay in the clinical chemistry lab. So total protein. When you say total protein class, this is the totality of all the proteins found in the plasma, in the specimen, the blood specimen. Uh, Proteins in the plasma are mainly synthesized by the liver. Okay? The liver is the organ where synthesis of proteins take place. All right? Now, when it comes to immunoglobulins, though, for the antibodies, they are synthesized by a specific type of cell called the plasma cells. Now, can you recall what are plasma cells? Have you taken your HEMA 1? Tapos yun ang HEMA 1 class?
Hindi pa po, sir.
Hindi pa. Okay. Have you met these plasma cells before? Familiar ba kayo sa plasma cell? Anyone? Wala? First time yung marinig tong plasma cell?
Sir, yung B cells po.
They are very good. They are B cells or B lymphocytes. Okay, somebody has remembered the anatomy. Very good. Okay, so plasma cells are actually B lymphocytes. But they are not just ordinary B lymphocytes. They are B lymphocytes that have undergone specialization or what you call further development. Okay, once the B lymphocyte becomes plasma cell, that plasma cell becomes capable of producing antibodies or what you call immunoglobulins, immune type of globulin. All right. So generally, all proteins are synthesized in the liver. For your antibodies, they are synthesized by a specific type of cell called plasma cells, which are differentiated and mature B lymphocytes. The metabolism of protein, particularly the amino acid part of protein, is done through a process called deamination. Okay? And in deamination, the amino acid part is converted into ammonia first, and then from ammonia, the body converts it into urea, okay, or urea. You want to read it as urea or urea. So urea class is a byproduct or a product, sorry, not byproduct, but a product of protein metabolism. Okay? Or you could also say amino acid metabolism. The urea is the waste product. Okay? Ammonia is the first type of waste created, but that ammonia is converted by your liver into eventually urea. Okay? It doesn't stay as ammonia. It's converted into urea. If your liver is functioning well, the ammonia has to be converted into urea. If the liver is diseased or is not functioning properly, then that's the time that you could see accumulation of ammonia because the amino acids are not anymore completely metabolized to form urea. There are also some proteins called cytokines which are produced from the site of injury or inflammation by immune cells, okay, like your lymphocytes and other white blood cells. They produce these cytokines, okay. The cytokines, when they are secreted by inflammatory or by these cells, the cytokines will stimulate the liver to release a certain group of proteins called acute phase proteins. These acute phase proteins class are produced when there is an autoimmune disorder, infection, tumors, acute myocardial infarction. Okay, in AMI, the myocardial cells die as a result of cell death. APP or acute phase proteins are released. Okay, as well as trauma or surgical procedure. Okay, this will result to the formation of these acute phase proteins. Now, when these acute phase proteins are being produced, there are also instances where the other proteins in the body suddenly drops or are not synthesized. We call this as negative acute phase proteins. Okay. Negative acute phase proteins means that during injury, when acute phase proteins are actively being produced, the other proteins like prealbumin, albumin, and transferrin, their synthesis is decreased. Okay, that's why they are referred to as negative acute phase proteins. The reference range. This is only one in almost all references. This is consistent. The total plasma protein is between 6.5 to 8.3 grams per deciliter. Per 100 ml of blood, it should contain 6.5 to 8.3 grams of protein. The unit class is in grams, not milligram. It's gram. Okay? If you want to express it in grams per liter, you just multiply this by 10 because in 1 liter, you have 10 deciliters. 10 times 100, that's 1,000 ml or 1 liter. So 65 to 83 if it's grams per liter. If it's grams per deciliter, it's 6.5 to 8.3. That's the reference range, or in the past, we call it as normal value. Okay? Now, if the value of the total protein is less than 6.5, the person is said to be suffering from hypoproteinemia. Hypoproteinemia. When do we usually see this hypoproteinemia? We have urinary loss of protein. Okay? There is a leakage of protein in your urine, may be brought about by damage to the kidneys. Okay? Also, gastrointestinal tract inflammation, liver disorder, malnutrition, inherited immunodeficiency disorder, and even in cases of extensive burns. Extensive burns will not just destroy the body physically, but also the biomolecules like proteins are depleted. Okay, resulting now to low protein in the blood below 6.5, or what you call as hypoproteinemia. If the protein is above 8.3, the person is said to be suffering from hyperproteinemia. Okay? The usual cause of hyperproteinemia is dehydration. Usually, a hyperproteinemia and dehydration is a result of depletional hyperproteinemia. Depletional meaning, hindi naman talaga tumaas yung protein level. Yun nga lang kasi nabawasan yung water level. So, yung ratio between the solute and the solvent has been disrupted. Okay? Mas marami na ngayon yung solute kaysa sa solvent. So, parang na-concentrate yung solute. That's why there is hyper or increased level. It can also increase through increased protein production. Okay? So magkakaroon din yung patient ng hyperproteinemia kung talaga namang mayroong increased protein production. So it's the opposite of depletional. Talagang tumaas yung protein production, like in cases of monoclonal and polyclonal gammopathies. Okay? Like in the case of multiple myeloma, for example. Okay? That can result to an elevated protein level. And even chronic inflammatory diseases that are associated with the production of paraproteins. Okay? That's for total protein. Now, in the lab class, when we receive test requests involving protein, so it's usually abbreviated as TPAG. Okay, the doctor would just write serum TPAG. What is that serum TPAG? It's serum total protein, albumin, and globulin. Okay, so you'll have to measure the total protein as well as the albumin. The globulin is a derived value. Okay, we assume that total protein is mainly made up of albumin and globulin. So naturally, after natin ma-measure ang albumin, like total protein, we can detect already the globulin by just deducting the albumin from the total protein. Okay? You might say, how come you don't account or take into account, sir, the other proteins? Well, the other proteins are grouped into globulin. Okay? The globulin group. All right. So, um, we now go to the test methodology for albumin. We have a dye binding method called the bromcresol green or bromcresol purple. Okay. Just like other proteins, the albumin can bind to an anionic dye, a colored substance, forming the albumin dye complex, which will shift the maximum absorbance of that colored substance or dye. And the shift or the increase in absorbance is directly proportional to the concentration of the albumin in the sample. Okay? So the more intense the color of the final product, the higher you'd expect the concentration of the substance. Right? So the reference range for albumin is between 3.5 to 5 grams per DL. Grams per DL. Okay? For the globulin, you just have to deduct the the albumin from the total protein to get the globulin amount. And then when we are requested to perform total protein or protein analysis in the serum, we are also required to compute for the albumin globulin ratio. So albumin globulin ratio is just dividing the albumin level that you measured with the globulin that you were able to obtain from deducting albumin from total protein. Okay? And the ratio will tell you important information as to what could be potentially wrong in the body. Okay? Now, a quick notes for the other related matters on protein. Um, we also have what we call a prealbumin, which is also known as your transthyretin substance. This prealbumin or transthyretin is an indicator of the nutritional status of a person. Okay, this is also a protein that is involved in the transport of thyroid hormones. Okay, then for the albumin, albumin, like all other proteins, are synthesized or is synthesized in the liver. It is the protein which has the highest concentration of all plasma proteins. It can also serve as a transport protein, and it's the main protein that contributes to the plasma osmotic pressure, which controls the movement of water in the body. Okay? Are there any questions or clarifications? Anything that you'd like to ask? Ah, point 3. Yes. 6.5 to 8.5. Sige, we'll accept that range then. Anything else? May tanong pa, class? So we will accept 6.5 to 8.5, okay? Up to 8.5.
Yes,
Anyway,
sir.
it's just point to discrepancy.
Sir, clarify ko lang po sa may total protein.
Yes,
Ang
Mark.
mini-measure doon, sir, is the albumin and the globulin, sir.
All, yes. Mainly the albumin and globulin.
Oh.
Kasi sa ating blood class, I will stop presenting. Mag-open cam nga kayo kasi hindi ko na kayo nakakita masyado. Hindi ko na alam kung ano mga itsura ninyo. Open cam. Can you please open your camera so that I can see your beautiful faces also? Yung hindi mag-open cam, absent. Sino nagsasabi?
Sira po yung...
Kaya man tira rason yun lang kay taglukat ti camera yun, O, Mr. Briones, paano kung malalamang nandyan kayo? The 21 na nga lang tayo sa class. Hindi kayo nagpa-participate class, nag-observe si Ma'am Sheila. Wala kayong participation, cooperation. Si Mark lang ang nagtatanong. Di ba lang kayo nagpabida para at least ma-impress si Madam? Tuloy nilayasan niya tayo. Wala na si Ma'am, no? Umalis na si Ma'am?
Sir, andito po ba si ma'am?
Oo, nasa class siya kanina. Mas nauna pa nga siyang dumating sa inyo.
Hindi ko po alam.
Kaya ako tawag na mag-recite. Wala man lang nagpabibo. Yapo. Hindi ba kayo pinaggatas ng mga nanay ninyo? Anong pinainom sa inyo ng mga baby kayo? Alam niyo kung ano yung Mr. Biones, are you present or not?
Sir, naka-open po pero ayaw pong mag-open. Sir,
Ang tanong, Mark, yung tanong mo kanina is total protein.
Sir, mainly ang mini-measure is are the albulin and globulin, sir.
Lahat na yon. So total protein, lahat ng protein na doon na fraction. Yun nga lang kasi, as we have said, total protein or the main proteins in our body are albumin and globulin. Okay? So when you say total protein, you're measuring both those two and the subtypes. But the subtypes are, when it comes to the amount, they are, well, we cannot really say insignificant. Okay? They're still significant, but quantitatively, amount-wise, talaga ang magmamatter lang is yung albumin tsaka globulin ninyo. Okay?
Yes,
Maliwanag? Any update pala before tayo, tapos na ang lecture, by the way. So, thank you for participating. Do you have any questions? May tanong pa ba?
sir. Sir, ay,
Sige, Mark.
wala na po pala, sir.
Miss Morty. Hindi ko alam kung ikaw yung nandyan or Momo. Nagsignat mo tayo, mabasang ko. Sino sa inyo ang hindi nakakaintindi ng Ilocano? Ilocano kay Ilocano, kayo magkamakawawat. Sino ang hindi nakakaintindi ng Ilocano? Everybody understands? Wag, paiso. Kasi ni Miss Ramos, hindi siya nakakaintindi ng Ilocano. Angeline, can you understand Ilocano?
Yes, sir.
Okay. Mukha ka kasing foreigner, naning. Nagpo daw ka. How about Mr. Briones?
sir, makaawat ako.
Hindi man lang magpakita.
Sir, naka-open cam po talaga ako. Kahit send ko po
Bakit
sa
na
GC.
nakikita namin yung picture mo? Miss Lopez, do you have anything to say? Umani-smile ka dito, di ka nagsasao.
Man, sir.
Mr. Andre Kilang? Is it Kilang? Si Jovi? Andre, ano yung first name mo? Jovi or Jove?
Jove po, sir.
Jove, okay. Before we go, can I just ask, how are you doing with your project? Nakasimula na kayo? Submission niya sa next Friday, sa May 20. 20, no? Tapos, pag nag-submit kayo, you email a copy to everybody para may kopya yung mga klase ninyo. Are we clear?
Yes, sir.
All right.
Yes,
so,
sir.
thank you. we have one topic left, which is non-protein nitrogen, NPN. Ano nang ginagawa nyo ngayon sa lab? Sa chem lab?
Tapos na namin yung uric at saka BUN, sir.
So, nag face-to-face kayo para sa BUN tsaka uric? Marigold?
Yes, sir.
How about yung sa total protein, nag-perform din kayo?
Sir, sa amin po hindi. Pinadrawing, sir.
Bakit hindi ay? Di ba lahat kayo is required na ng F2F?
Sir, ubos po yung reagent na sir, kaya hindi po namin ginawa.
Wala kayo. Ang tagal-tagal lang nilang hindi nagpalab, 2 taon, tapos nung nagpalab sila, wala, ubos ang reagent. Nyamot turn. Anyway, so kailan ang kwan? By the way, class, ang exam ninyo, ang final exams will be on campus. Pero yung exam questions will be delivered to you via the LMS. So yung exam ninyo is via online pa din ha. Doon lang kayo sa SMU campus mag-exam. Naintindihan? So during the exam schedule, kasi ang exam schedule ngayon will be common schedule, lahat kayo sa isang oras. Dalhin ninyo yung inyong laptop. And then I also would advise you to bring, I don't know kung meron ba kayo dyan, pocket wifi? Pocket wifi ba yung mga ginagamit nyo? Or data?
Sir, data lang.
O paano kayo mag-exam sa SMU? Malakas ba ang signal sa SMU?
Sir, meron po yung ibang part ang hina po.
Ala, kasi ano kayo, panda kayo di classroom nga, awan ti signal na. Because you will all be... Taga saan ka ba, Miss Alessa? Hindi, lahat kayo require na pumunta sa campus. Lahat kayo pupunta sa campus. Meron ba ditong taga outside Nueva Vizcaya?
Meron, sir.
Sino?
Yes, sir.
Ako, sir, Isabela po.
Marigold, hindi ka pa pupunta ng Bayongbong simula ng pandemic? Hindi ka pa nag-join ng F2F, Marigold?
Aman, sir, pero umuwi po. Kasi mahina rin po yung kapag sa boarding.
Ay, so. Alam, paano yan ay? Hindi pa ba? Oh, anong sabi sa inyo ni Sir Justin, Alisa? Oo, yun ang sabi. Ang sabi namin actually is online, pero yung delivery lang yun ng question online. Pero yung venue sa campus, kailan sinabi sa inyo ni Sir Justin? Oh, nag-meeting kaya sila? Hindi ako nasabihan Sige, tanungin ko na lang kay Ma'am Sheila and then i-finalize ko sa inyo sa Friday next week. I-remind nyo ako, okay?
Sir,
Yes, Mark?
kailangan pa ng camera.
Kung sa exam is hindi kayo i-require pumunta sa SMU, kailangan nyo ng second camera. Pero kung sa SMU kayo mag-exam, may proctor na doon. So, not necessarily na mag-camera pa kayo, pero kailangan nyo pa rin internet connection kasi yung questions nasa LMS ninyo. Saan ka ba located, Mark?
Sir, taga Isabela po ako pero andito na po ako sa Bayombong, sir.
May boarding house ka dyan?
Meron po, sir.
Walang, pero okay naman ang wifi.
Yes, sir. Okay naman po.
Oh, okay. Sige. All right, class. If there are no more questions or clarifications, thank you very much for taking your time to attend the class today. Most of the people from Bayombong, I think, were not able to join because of the power interruption. But they should not worry because the lecture today has been recorded. So as soon as ma-download ko yung recording from Google, I will send you again the recording links. Nabubuksan nyo ba yung mga recording links na sinisend ko sa inyo? Okay. Sige na nga, Rod. Thank you very much. Pag wala nang tanong, you can go. Have a wonderful evening. Stay safe. And I'll see you again on Friday. By the way, dahil na-observe tayo ngayon, hindi ako nakapagpa-quiz, I will be giving you 2 long quizzes next Friday. Lipids and proteins. Okay? Do not forget, all multiple choice, lipids and proteins. Are we clear? I will upload the lecture notes and the recording and the PowerPoint in the LMS. So just check your LMS. All right? Okay. Bye-bye.
Bye, sir.
Bye,
Bye,
sir.
sir.
Bye, sir.